Good afternoon, good morning, good evening, everyone here in this webinar on patient preferences to assess the value in gene therapies, which we call the PAVING study, and which is a study of the IMI PREFER project. Welcome, everyone. We will give this presentation with three experts of the PREFER consortium. First of all, Eline van Overbeeke, who is Health Economics Outcomes Officer at Pfizer at the moment, but during the moment of the study was working at the KU Leuven. Brett Hauber, who is also working at Pfizer and who is Patient Preference Director at Pfizer, but who was working during the patient preference study at RTI, and then myself from the University of Leuven in Belgium. Before we go further with the real content of this webinar, I give certain tips and tricks and aspects what you could do in order when you have questions. So submit your questions in the chat tool, which is available on your screen, or raise your hand and voice during the question and answer session, which we will organize at the end of this session. So we will have 15 minutes questions and answers. And if you would have any other question after the webinar, please feel free to contact the PREFER consortium via this contact email address or by directly contacting the speakers of today. This webinar is recorded. The recording will also be made public afterwards. The session on the question and answers afterwards, so this 15 minutes slot, will be cut out and so will not be published. PREFER has established a series of webinars with all sorts of topics that we have been covering in PREFER and which are available on the YouTube channel with the link here present on the slides. There are indeed certain other webinars which are upcoming and for which I also encourage you to participate. You're very welcome to listen to these other topics. So today we have an agenda where I will present the topic of today, I give an introduction about decision-making and the clinical context of the topic of today, namely gene therapies. And afterwards, Brett Hauber will give you the design of the patient preference study. After which Eline van Overbeeke will give you insights into the results and also draw some conclusions about this PAVING study. Brett and Eline are currently hence employed by Pfizer. So at the moment of the study, they were not employed by Pfizer. This study was completely funded or sponsored by IMI PREFER, which is the Innovative Medicines Initiative, not receiving other financial support from other sources. And the pharmaceutical companies, which are also involved in the project, were in this case study involved in the advisory board, which extended the team, and they had no access to the data itself, so we exchanged views with them. And the researchers were not influenced by any pharmaceutical company in this aspect. So all the views which will be presented in this webinar are the views of the individual presenters and should not be linked or to any organization that the speakers are linked to. Okay, so let's give this introduction for today. We are talking today about patient preferences, more specifically in the context of gene therapy. And why gene therapy? Because that causes and may generate potentially in the future a huge promise to prevent, halt, or cure diseases that are genetic in origin. And what does it do? It may alter the genetic material in the disease-causing cells of the patient itself, or it may do this in other cells of the patient in order to correct the activities of the disease-causing cells of the patients. We can think of the CAR T-cell therapies. Or it may be that cells are genetically engineered outside the patient, which may be then provided to the patient to correct the activities of indeed disease-causing cells of the patient. And that type of intervention may produce permanent effects in the patients, which indeed also contributes to the big promise that gene therapy may give, potentially and hopefully also for patients that suffer from haemophilia, because that is indeed the disease context, the clinical context that we will focus on. Haemophilia is a genetic disorder, and this means that the disease is caused by an error that is situated in the DNA of the gene of clotting factors. And if that is in clotting factor 8, then we call it haemophilia A. If it is in clotting factor 9, we call it haemophilia B. And this clotting factor is crucial to stop bleedings in normal situations, as it helps the blood clotting. However, when patients have haemophilia, the clotting factor is defect. And in this way, indeed, the error caused in the gene produces either a missing protein or a defect protein, which means that the bleeds cannot be stopped. So the bleeds are longer and often occur spontaneously. The standard therapy for the moment supplements the body with the clotting factor itself. And now there is a new therapy, namely gene therapy, in development, which corrects the error in the genetic material of the clotting factor, so that the body at that moment has the correct gene to produce this missing factor. And as I told you, this gene therapy is still in development, and such development goes over clinical trials in order to produce the necessary evidence for regulators and for payers to make an assessment about the market access. However, there are, for the moment, certain benefits and risks which are uncertain on the short term, uncertain on the long term, that needs to be further assessed. These uncertainties also needs to be taken into account in order for the regulators and for the payers to decide about marketing authorization, pricing, and reimbursement. And it is very important also to understand whether or not patients are able to accept uncertainties related to gene therapies. And what other important aspects are related to gene therapies that need to be taken into account when talking about the value of such gene therapies. And this is what PREFER and actually the PAVING study aims to perform, namely, in this choice between the standard of care, the prophylactic factor replacement therapy, and the gene therapy, what are the features most important to people with haemophilia? Can we identify them, and can we also quantify them? And what are the trade-offs that patients with haemophilia are willing to make when making that choice? This is a study which has been executed in a collaborative setting between the sponsor, which is the IMI, and more specifically the PREFER project, and then the study team which collaborates in this IMI project, namely the University of Leuven, KU Leuven, and RTI Health Solutions. Very important was the advisory board, where we touched base with several stakeholders, the HTA payers with the KCE of Belgium, Zorginstituut Nederland, from the HTA payer point of view, the regulators, the representatives of the European Medicines Agency. We had also a representative of the industry, namely Pfizer, clinicians from the university hospitals of Leuven and also Saint-Luc in Brussels, and patient organizations were also involved in this study, namely from the EUPATI, Eurordis, and the AHVH organization, for which we are very thankful that they contributed a lot in this study. And this brings us to the design of the patient preference study, where I give now the word to Brett Hauber, who can give you more insights into that.
Thank you, Isabelle. So I'll describe the process by which we developed the study. And the methods here were pretty rigorous. We started with a literature review that looked at both the preference elicitation method and the identification of the treatment attributes. And then key to the entire process was the advisory board that Isabelle just described. One of, I think, the biggest strengths of this study was having such a diverse advisory board throughout the process that helped us ensure that we were staying on track, answering the questions that mattered, and including the features that mattered not only to the people with hemophilia, but to the decision makers as well. Prior to initiating the survey, as part of survey development, we did 20 interviews in Belgium with people with hemophilia to look at what information needs they had and their understanding of gene therapy, the factors that would influence their choice of treatment for hemophilia, including gene therapy, and then we identified the top treatment features for hemophilia, assembled these together, and then we administered the survey to 117 people with hemophilia in Belgium using the threshold technique. So as an overview of both of these phases, the first method included semi-structured interviews with both open questions and a ranking exercise, the objective being to identify the treatment features that mattered. There were 20 interviews among people with both hemophilia A and hemophilia B, most with severe disease, and this comprised 4.5% of the population of people with hemophilia in Belgium. And for the full survey, it was a fairly standard patient preference survey looking at including demographics, disease and treatment history, quality of life, and a preference elicitation technique. But one of the things that was unique about this study was it included an interactive educational tool, which I'll talk about in a moment. And the goal was to look at what the trade-offs were that people with hemophilia would be willing to make when considering a gene therapy. And the survey was 117 people, which is on the smaller side for patient preference studies as a whole. But it's important to note that this is a rare disease, and this comprised 26% of the population of people with hemophilia in Belgium. So I had mentioned before that one of the unique aspects of this study was the educational tool. And the educational tool was actually kind of fun to see. It was an interactive tool, but it was developed in a very deliberative process. The goal of this was really to answer a research question that IMI PREFER had put forward, which was, what is the impact of educational tools on outcomes in patient preference studies? So we did a literature search to understand not only the information needs that were important, but also how to structure the tool. We had interviews with patients. Again, remember we talked about information needs. That informed the tool. We asked hematologists to validate it for accuracy and to ensure that we weren't missing anything. We asked the patient representatives on the advisory board to help us make sure that it was correct, and then we piloted it. The tool itself was animated, as you can see here, and it was interactive. The animations were actually not static, they were dynamic. There was a voiceover to help people understand and not only have to read, and it covered 5 different modules so that people would understand how gene therapy works, what it generally does, how it is administered, what the effect might be, and what the side effects and uncertainty might be. So the next question that we had to answer was, for the full survey, which was the method that we would use? So again, this followed a fairly deliberative process. We started with 9 promising methods from the work that PREFER had done. We looked at that in relation to the research question, the relatively small patient population, the decision context, and the requirements that we were going to want to impose to establish the validity of our results. We then sought expert opinion on methods and came up with 2 methods that would be worthwhile or promising in this particular case. One was the threshold technique, the other was swing weighting. And in the end, we chose threshold technique for the fact that we can do it with fewer people, knowing that this was a rare disease. This was going to be an unsupervised survey, which made swing weighting somewhat problematic. And another thing that we'll see when we show you the questions is that this was a labeled design, meaning that we actually compared gene therapy to factor replacement as opposed to having an unlabeled design, which is common in most patient preference studies. Then we had to choose the attributes. So we started with a literature review. We had information and input from the advisory board. We tested those thoroughly in the 20 patient interviews and brought them back to the advisory board for clarification, because we had, as you will see, 5 top attributes, but there is confounding between factor level, bleeding rate, and the probability that prophylaxis could be stopped with gene therapy. So we had discussions with the advisory board about how to structure these 5, and in the end it was determined that we would use 4 instead of 5. In other words, that we would not include factor level. So the survey looked like this. And the first question in a threshold question is what we could consider a direct elicitation, meaning that if these 2 options, prophylactic factor replacement therapy and gene therapy, are described in such a way that they represent a potential reality, this actually would tell us that given those options, which people would choose which therapy, and Eline will show you some of those results. So this is our starting point, and everybody started at the same place. Again, we talked that they were labeled. A is always prophylactic factor replacement therapy, which is standard of care, which is typically the left-hand side in a threshold technique. Gene therapy is the alternative. In this case, again, labeled as gene therapy. The primary differences in where we started was that the likely benefit would be a high probability of being able to stop prophylaxis. The risk of gene therapy in this case was that we had fewer years of information, so we had much more uncertainty about side effects. To be conservative, we assumed at baseline that there would be 6 bleeds per year in both factor replacement and gene therapy, and that the average quality of life reported in the clinical studies would be the same between the 2. So this was our starting point, and we were able to then say, given gene therapy with this particular profile, would you choose this over factor replacement? And if someone did choose gene therapy, in this case, it told us a few things. First, that gene therapy was an acceptable alternative. There will be some people who would not even consider gene therapy under any circumstances. It also told us that the probability of reducing the need for prophylaxis outweighed the uncertainty surrounding the side effects, given the fact that we had fewer years of time during which it was studied. And again, the bleeding and the quality of life were the same. Once we established the baseline, we then adjusted each of 3 of these one at a time to see where the threshold was at which someone would switch. So if someone chose gene therapy to start, we knew that we needed to make gene therapy look worse in order for someone to switch back to prophylactic factor replacement. If someone chose prophylactic factor replacement first, we would need to take gene therapy and make it look better to entice them to switch. So first we did bleeds per year. We then did the chance to stop prophylaxis, and then we looked at changes in quality of life that would be required, all as offsets to the uncertainty of having less information about the side effects that were studied. And with that, I will turn it over to Eline to describe the results.
Thanks, Brett. And thanks, Isabelle, as well, for the great introduction. So let's have a look at the study results, as Brett already mentioned. Now, we focused a lot on the baseline profile. As Brett mentioned, 6 bleeds per year for both therapies, 90% chance to stop prophylaxis after using gene therapy, no impact on quality of life, and only 10 years of side effects follow-up for gene therapy. What we saw here is that already 61% of patients would prefer gene therapy over their standard of care in this case. So this already told us that there were quite a lot of patients willing to use gene therapy. But let's have a look at the 3 attributes that we changed throughout the survey. First of all, the annual bleeding rate. So here the question was at how many bleeds per year a patient doesn't have a preference anymore between gene therapy and their standard of care. So the rest of the levels stayed the same for the other attributes, but we were really looking into the bleeds per year. Now, when we have a look at this figure below, it shows you the percentage of patients that would prefer gene therapy at the different levels of bleedings per year. And when we look at the baseline question, we here see again the 61% of patients that would prefer gene therapy over standard of care if the number of bleeds are the same with both therapies. So that's our starting point. Now, when looking at the left, we also see the percentage of patients that would prefer gene therapy when gene therapy would come with 0 bleeds. And this is 88%, so quite a lot. But there's also still a number of patients that would never want to use gene therapy under the conditions presented here. So even if gene therapy would result in 0 bleeds, then they would still not accept gene therapy. And that was 12% in our case here. Then looking at the next series, so chance to stop prophylaxis, similar to the annual bleeding rate series, here we wanted to see at what percentage chance to stop prophylaxis patients would no longer have a preference, so to see when they would switch. Looking at the figure below, we see again then the preferability of gene therapy according to chance to stop prophylaxis. When we start here at baseline, so 90% chance to stop prophylaxis after gene therapy, we see again the 61% of patients that would prefer gene therapy. When we then look at the left, when it's going towards 100% chance to stop prophylaxis, we see that 68% of patients still wants to receive gene therapy. But here we see also a higher percentage of patients that would not want to use gene therapy in this case, in this profile as shown above. So this told us that probably the other attributes like annual bleeding rate are a bit more important for them to consider when making their choice for gene therapy or not. Now, when we look on the right side of this graph, we also see that there is still a small group of patients, 29%, that would still prefer gene therapy, even if there is a very low chance to stop prophylaxis, 20%. And in interviews with patients, we saw that these are patients that would accept any type of chance to stop prophylaxis, really to get a break from that continuous use of prophylaxis therapy. And then the quality of life series. So here the question was regarding the level needed for quality of life to switch. We will start again at the baseline question. So here we see again that 61% of patients would prefer gene therapy if there's no difference between quality of life with the standard of care and gene therapy. When we go down to the left, we see that there is 87% of patients that would prefer gene therapy if they would be able to get almost perfect quality of life with gene therapy. There's also still a group of patients that would not accept gene therapy, even if it could result in 100 quality of lives on a score of 0 to 100. So this was 13% in this case. Now, as you saw throughout the graphs, there was quite a lot of preference heterogeneity. Some patients needed more benefits than others. And these are the factors that we found that impact that acceptance, so that heterogeneity that we saw. First of all, regional differences. This was one of the most important factors. We saw that participants living in Flanders, so in the north of Belgium, required less benefit than those living in Wallonia, so in the south, to prefer gene therapy. But then a second runner-up was time on educational tool. And we saw that the more time participants spent on the educational tool, the less benefit they require to prefer gene therapy. This sounds maybe a bit strange, but it's actually quite logic, we believe. in line with also the Rogers diffusion of innovation theory, where we see that we need education before we can accept new innovative concepts. And then we also have two other factors that contributed to this heterogeneity, or were able to explain it a bit, but only in some of the series that we did. So age, the older the participant was, the more chances of prophylaxis they required. So that only had an impact on that series. And then also their current annual bleeding rate had an impact on the annual bleeding rate they would want to have from gene therapy. So those were the results, but what does this now mean in practice? We, of course, set this study up from a market access perspective, so HTA and payers. And here we think that we have a lot of ways that we can use these results of this study. On the one hand, when we look at the top treatment features that we were able to identify in the qualitative phase with the interviews, we think that this can be helpful to design outcome-based agreements to make sure that the outcomes included here are also relevant to patients and in a similar way in real-world evidence studies. And then regarding the trade-offs and acceptability, so the results that I just showed you, we think that this may be helpful in valuation of the benefits, the risks, but also other elements that go beyond health gain. And then last but not least here, these results can also help to get a better estimation of the budget impact of gene therapies. So matching the gene therapy profile to the percentage of patients will show us how many patients will actually accept gene therapy in real life. And this may be lower than what we would expect. So in conclusion, we have identified the top 5 of attributes, of which we only used 4 in the survey, but the top 5 was annual bleeding rate, factor level, uncertainty and long-term risks, impact on daily life, and probability that prophylaxis can be stopped. And when used in the survey, we were able to identify the willingness of patients to receive gene therapy. And up to 88% of patients with haemophilia would prefer gene therapy over the standard of care, at least in the conditions presented in our survey. In contrast, at least 8% of patients would never want to accept gene therapy. So these are the patients that across the different series, so across the questions regarding annual bleeding rate, chances of prophylaxis, and quality of life, would never accept gene therapy. And this was 8% in our sample. We also identified preference heterogeneity as shown, and we believe that this is something that should really be explored in great detail in future preference studies on gene therapy. And we then also saw that the educational tool can educate patients effectively and improve gene therapy acceptability. So with that, I think we can finish the presentation. I would like to thank some people that helped us setting up this study, as you can see here. Thank you for your attention, and I think we will go to the Q&A then.
Yes.